So my name's Louisa and I'm a chemical content curator for the ChEMBL team. And with me I've got Anna, who's our data integration officer, and Prudence, who is our bioactivity curator. And Prudence and Anna will be answering any of your chat questions that you have. So firstly, we'll start off with what is the ChEMBL database. So for those of you who don't know, for those of you who haven't used ChEMBL before, we are known, or we're seen as, open access data for drug discovery. The core of our database has been extracted from the primary medchem literature, and it covers at least 33 years of research. The main journals that we extract from are JMedChem, Bioorganic Medchem Letters, and Science, but we do have approximately 47 different journals that we extract from. We do have a data sharing arrangement with PubChem, and from PubChem we take their confirmatory and their panel assays with the dose response endpoints only. We do also submit into PubChem ourselves, so they take our data and we take theirs, but everything is annotated, so you will always know where the data has come from. We do also have contributed data sets, so we have neglected tropical disease data, which was donated very kindly in 2010 by GSK, so that's approximately 13,000 compounds and associated activities. We also have the donated GSK kinase set, data from TG Gates, DNDi, Novartis and Harvard, to name a few. We also annotate and store marketed drugs. We take these mostly from the FDA Orange Book and Daily Med. We understand that there are other places that we can get approved drugs from, but we choose FDA as an arbitrary source. The data content that we have is updated regularly with data. We aim to do a release roughly 2 to 3 times a year, depending on resourcing. All the bioactivity data that we have is associated with a chemical structure, a biological target, which can be a protein, a cell line, an organism, and we'll also give you the provenance. So you will always know where that data has come from, whether it be PubChem, a deposited set, or extracted from the literature. The chemical structures and the biological data are curated in-house to ensure quality. So this means that once it's been extracted and put into ChEMBL, it doesn't just stay there, it is constantly checked and curated as it's needed. We also have links to key EBI and external resources. So on our interface you will find links to other EBI resources such as Reactome or Interpro, but we also have links to external resources such as UNIPROT, PWE, World Health Organization, etc. The ChEMBL interface is searchable by chemical structure, by keywords, and the keywords can be linked to targets, assays, documents, or compounds, and also you can search by target sequence. We do have secure searching using HTTPS, and this is because we understand how important security is when you're using internet resources. So we will never store any of your searches, we will never store any of your personal information. The only thing that we will store is where you are in the world and the fact that you've clicked on ChEMBL. We also provide downloads of the ChEMBL data in various formats, so depending on your system, you can download SD files, text files, Oracle dumps, or other database dumps. Just to run through some stats of what we currently have in ChEMBL, approximately 92% of ChEMBL are made up of small compounds and about 7% are peptides. The other 1% are made up by anything that cannot be classified as a small compound or a peptide. We've currently abstracted data from 59,000 papers across 47 different journals. This has meant that we have 1.7 million compound records, with about 500,000 directly from PubChem. We have 1.4 million distinct compound structures, and from this we have 13.5 million activity points, of which 6 million have been directly extracted from PubChem. And we're currently looking at storing binding, functional and ADMET data. We have over 10,700 targets, of which 5,400 are protein targets and over 2,400 are human targets specifically. And we have just over 1.1 million assays in total stored in ChEMBL. Okay, so just to give you a quick ChEMBL extraction overview, what happens externally to our ChEMBL group is we have curators who will read through the papers. They will take out the target data, they'll take out the organism data, the bioactivity data and the compound data. This then gets sent to us separately, it gets loaded into the system, and it's at this point where we then curate it in-house. The target types that we currently store in ChEMBL, we don't just have a specific type, so we're not just interested in a single protein or protein family. We're interested in any target type that is published in these papers. So it can be a protein, or it could be an organism, or it can be a protein family or a complex. If the data has been tested against a protein complex, again, we will always tell you that it's a complex and not a single protein. So the targets are very highly annotated. Now we're just going to have a look at the ChEMBL homepage. Don't worry, the red box you will not see on the ChEMBL homepage. That's just to highlight certain parts that are quite useful. So if we have a look on the left-hand side, you will see that we have all the different subsections of ChEMBL. So not only can you click on ChEMBL itself and access this homepage, you can also click on Unichem, which is one of our other resources, malaria data, the neglected tropical diseases, ADME Safari, Kinase Safari, GPCR Safari, etc. I'm not going to go into detail about any of these today. These will be discussed in separate web conferences that you can link to, but I will be talking about ChEMBL and the downloads. Just further down, you have the ChEMBL statistics. So this is quite useful because it will always tell you what version of ChEMBL you're currently using on the interface. It also tells you how many compounds to expect, how many activities, and how many targets. Another thing that's quite useful is our ChEMBL blog down in the lower left-hand corner. So this blog is handy to keep up to date with what's happening in the ChEMBL group. So this can be a release of data, it can be a new way of searching, or it can just be when the next web conference is. So it's quite useful to click on that and have a look at what information is stored there. You can then see the main part of the homepage here is focused by default on compound searching. So we use Marvin by default, and you can draw in your structure of interest and click on a substructure search or an exact match. Or you can run a SMILE search or a ChEMBL ID search, or you can run a BLAST search on the biologicals. However, I personally think the most important part of this interface is this top field up here. So this is where you can use simple words, keywords and wildcards to search across compounds, targets, assays, documents and cells in ChEMBL. So if you don't quite know what you're looking for, you can put a partial name in here, use the asterisk and search across any of these subsections. So we'll now go into the interface searching. So as I mentioned, you can use keywords. So when searching for a keyword in compound, you can look at trade name, synonym, or systematic name, to name a few. If you're looking across targets, you can have a look at a target pref name, so ERBB1 or ERBB2 is an example, or you can even use a uniprot accession such as P11229, or even a partial name of a target. Same with assays, you can look for a keyword, so you could look for a cell line with an assay keyword, or you could look for hepatotoxicity, or even an organism. You can also search on structure. Again, I mentioned you can do this by substructure, or you can do it by an exact match if you know exactly what you're trying to find. Or you can use a BLAST search. So this is where you can go and download a FASTA file of a target, and search through ChEMBL using this to either find the target itself or something that has a sequence similar to your target of interest. So just to go back to the home page, I mentioned that you can use keywords up in this field at the top. This is very useful. You can draw in your structure here and you can run it as a substructure or a similarity, or if you're more chemistry minded and you know about smiles, then you can run a list of smile searches here. I believe this is restricted to about 1,000, but you can run quite a long list of keywords or smiles or ChEMBL ID of compounds if you know what you're trying to find. So once you've run your search, you will be brought back the search results. So this will be in this grid form, and each of these columns are audible by clicking on the small arrow at the top right-hand corner of each column. You can see that you have each structure in the left-hand side, you have known synonyms for the structures, and then you have the compound properties of the compounds. And then up in the top right-hand corner, this drop-down box will allow you to download the data as bioactivity data or an SD file. If you choose to display the bioactivities, you'll be taken to this screen. So this is where the bioactivities are again in a simple grid form with the compound to the left-hand side. You will then have access to all the activities for those compounds, the assay data. All of this is hyperlinked, so if you want more information about the assays, you can click on the hyperlink. It will tell you where the data is from, so in this case it's scientific literature, but it could be from PubChem or a deposited set. It then gives you the target data, so you'll see here that we have it linked to a single protein, we have no relevant target here, and we also have a cell line. We then have more information about the target in this column, the organism, and then you have links to the references. So as I mentioned before, the provenance of all the data will always be shown on the interface, so you can always see where this has been extracted from. I'm now just going to have, actually, before I do have another poll, I'm just going to show you can also download the data to an Excel file. So all the data that you've just seen on this screen would be transferred into an Excel file, which can then be manipulated, it can be used in other programs, and it can be mined for useful information. You can also download the structures as an SD file, so again, if you're more chemistry minded, more interested in the structures, you can download the data as an SD file, and you'll see that you have the compound information in there as well. Okay, so just to give you a bit more in-depth about the data that we do store in ChEMBL. So I've shown you how to do a very simple search on the interface. But what you can then do is, if you're interested in a specific compound, you can go into what's known as our compound report card. So this is where we store and we show you all the information that we've gathered for that compound. I've chosen lapatinib, which is a kinase inhibitor, as my example compound. And I've laid it out on this slide, not in the same way that you will see it on the interface, but it's just so that you can see things side by side. So you'll see that on the compound report card in the top left-hand corner here, we have the compound name and the classification. So you will see that we have a ChEMBL ID, so ChEMBL 554 is the unique identifier that we have for lapatinib. You'll see that we have the compound name as lapatinib. So this compound name generally is the associated registered name, so a USAN name, for example, which is the United States adopted name for that drug. We also store synonyms because we know that compounds can be known under many different types of names. So we have TICURB and GW572016F, which is the research code for this compound. You will also see that we've got a max phase, so this will then tell you whether the compound has been approved, which is phase 4, whether it's currently in clinical trials or stopped at a certain phase in clinical trials, which is 1 to 3. And if there's nothing in the max phase, then that generally means that the compound was the compound never made it to clinical trials. And we also have a formula because that's quite useful. Going a bit further down the page, we have compound representations. So we will store this compound as a mole file, as smiles, standard INCHI, and standard INCHI. So depending on how people store compounds in their own systems, these can be quite useful for them. For known drugs, we store their mechanism of action. So we know that lapatinib is active for ERBB1 and ERBB2, so we will have links to that target as well as the references where it came from. So if you click on the hyperlinks to the targets, then you'll be able to get more information about that target itself. These compound icons here, or the compound features, the green icons on the top level will tell you more about the compound itself, whereas the blue icons will tell you more about it as an approved drug. So the benzene ring in green just means that this compound is classed as being small. And in the blue icons, this will tell you that the pill says that it's by oral administration. The black box means that it has black box warnings, and the RX generally means that it's by prescription only. If you wanted more information about lapatinib in clinical trials, we do link to clinicaltrials.gov, the Cochrane collaboration, and the British National Formulary, and we do have hyperlinks out to those sites where you can get more information. We do store different forms of compounds, and by different forms we generally mean salts or isotopes, and we do link these together. So in this case we have lapatinib ditosylate, which is this compound. If you were only interested in the activity of the salts, then you can click on this hyperlink here. You'll be taken to a similar report card where the activities here will only be linked to the salt itself. However, being on the parent page, which is this one, the bioactivities here are linked to both the parent and the salt together. The bioactivity summary, this pie chart, this is where it splits out into the different activities that we have stored for lapatinib. So for lapatinib we have other, which could just be activities where the actual endpoint is not being specifically stated in the paper. We have IC50s, KIs, inhibition, activity, and EC50. If you were just interested in IC50 data, you can click on this as it's a hyperlink, or you can click on the red section of the pie chart because it's also a hyperlink. However, if you wanted all the data for lapatinib, if you click on this small grey box to the lower right hand corner of the pie chart, if you click on it, it will say download all activity data, and that will give you everything that we've got for lapatinib. Scrolling further down the compound report card, you'll see that we have target predictions. Now we know that lapatinib, being an approved drug, is active against ERBB1 and ERBB2, which is why we have this in our predictions. However, these predictions have been created from a training set, and we have this for every compound that qualifies in ChEMBL. So it could be that we have some compounds in ChEMBL that you've never thought of as being potentially good for a certain target, and we will have this on the compound report page, so you'll be able to investigate that for more information. We also have the calculated compound properties down here. So these are calculated rather than extracted from the papers. And the reason they're calculated is for consistency mainly. So when we have properties reported in papers, we don't always know the provenance of where those properties came from. So we use Pipeline Pilot and ACD Labs to calculate these properties on our behalf, and it means we have a consistency across all the compounds. I've put a red box around compound cross-references because it is important to say that we do have external links to as many different resources as we can. So for lapatinib, we've got links out to ATC, ChemSpider, PubChem. We've also got links to all of these Drug Bank, ChEBI, Atlas. So we really do link out to external resources wherever we can. So some examples of these are ChEBI, PDB, clinical trials, World Health Organization, PubChem, and ChemSpider. So a different type of search you can do is using a protein sequence search, otherwise known as a BLAST. So this is a more precise method for identifying targets, and what you input is a protein sequence of interest, and you do this by using a FASTA file, and this then has a BLAST algorithm performed on it to try and see if there's anything in ChEMBL that is similar or the same as the search you're currently running. We then score the results by percentage identity, so with the most similar sequence being at the top, going all the way down to the least similar sequence. So an example of how to do this is you find your protein sequence of interest. We normally use UniProt, so I've put in a partial name of the target that I'm interested in. I've run that search. I've realized that I want P11229, so I click on the target that I'm interested in. I will then go to the home page of that target. I will click on the FASTA download, and then I will copy this sequence here. I'll then go back to ChEMBL, and I will paste that protein target into the search box, and then I'll run the search. Once I've run the search, I'll then have the BLAST search results returned, and you will see that the query sequence that we've inputted is down the left-hand side, and then you'll have the percentage identity score over on the right-hand side. So you will see that the top 5 are 100% matches, and that's because they have my target of interest. So I searched across this M1 receptor for Homo sapiens, but we also have the complexes and the family linked to that target as well. And you will see that the next 2 that are very similar are the same target, but in 2 different organisms of monkey and mouse. So you can sort by any of these columns, but they're normally sorted by the percentage identity. So when you found the target that you're interested in, which is this one, you can then click on the hyperlink here, and you can go into the target report card. So for the target report card example, I've chosen a slightly different target, but the target report card is laid out in a similar way as the compound report card. So you will have the target name and classification at the top. So it tells you that it's a single protein. It will give you synonyms and a preferred name for this target, and all of these are searchable in ChEMBL, so you can use this full name or part of the names to search across ChEMBL. You will see that the organism is Homo sapien, and that we've classified the protein target as enzyme kinase protein kinase, TKL protein kinase group, and TKL protein kinase IRAC family. Target components for this, so this will actually tell you that this is a single protein, so there's only one target component. But if this was a protein family, we would list all the different targets that make up that family, and we have a link out to UniProt here. As with the compound report card, we have all the bioactivities that are linked to this target. So you can see we have KIs, inhibitions, IC50s, and like before, you're just interested in KI activity for the target, you can click on the KI hyperlink or the pie chart, or if you want everything, again, you can download it using this little grey box. Similarly, for the assays, you can just look at binding, or you can just have a look at functional. And then we also have target ligand efficiencies, and each of these dots relate to a specific compound in ChEMBL. They are clickable, they are hyperlinked, so you can get more information about the compounds, or as with everything, you can click on the grey box and download everything. Scrolling further down the compound report card, you can also access all the compounds that are linked to this target. So if you're not just interested in the activity, you want to see every compound that's ever been tested against this target, you can download them all here. Or if you're interested in a specific weight set, you can actually grey out different weights of the compounds and only focus on certain ones. As with the compound, we do link out to as many external resources as we can. So you can link out to CANSAR, which is run by the Institute of Cancer Research. You've got Intact and Reactome, which are internal EMBL-EBI resources, Interpro, PFAM, and we've also got the gene ontology as well. So everything is hyperlinked and everything is linked out to external resources. So going on to external resources, again, as with compounds, we've got loads of links out, so UniProt, PDBE, Intact, Expression Atlas, Reactome, and Human Protein Atlas. So now if we want to move on to browse targets, this is also something that's quite interesting to do because it allows you to look for families of targets or even organisms. So if we focus on the taxonomy tree at the moment, and you can find this browse targets from the home page. If you wanted to know all the data that we have for every primate target, all you would have to do is click on this checkbox here and then click on Fetch Selected Target, and that would bring you back a huge amount of data that's just purely focused on primate targets. Similarly, if you're just interested in fungi, then you can do the same. You can click here and download all the data or access all the data for the target related to fungi. The other side of the target hierarchy is the protein target tree, so you can click the radio button here and you can access all the different protein targets that we have. Because we do have a lot of them, over 10,700, you can search the tree by using a keyword. So in this example I use the keyword of membrane. So I typed in membrane, click search, and what this will do is it will expand the hierarchy tree for you, and it will highlight and make bold every section of the tree that contains the word that you've searched for. You can then click all the checkboxes to the left-hand side of each of these and get everything, or you can use the little arrows to the left-hand side to drill down further and really make your search much more specific. Once you've checked all the boxes you're interested in, you can then fetch the selected target and then have a look at the data associated to them. A useful thing that we've added, I believe, in the last release was the ability to click on these hyperlinks up at the top to do the searches for you. So, So we found that a lot of people were just interested in the protein kinases. And so rather than them having to look through the hierarchy, we've just created these hyperlinks which will do the search for you. This will take you directly to the page that lists all the different kinase targets that we have. Similarly for GPCRs, the ion channels and nuclear hormone receptors. If you can think of any other interesting subsets that would be useful for you, please feel free to send us an email. So that's how to search on the interface. So the other thing that we provide are downloads. So because everything that we do at EMBL-EBI is completely free, open access, everything that we have in ChEMBL you can download to your own system. So you can access the downloads from the home page by clicking on downloads. This will then take you to the downloads page, which will show you every subset that we provide of ChEMBL that you can download the data from. However, we're just going to focus on ChEMBL-DB. So if you click on the hyperlink for ChEMBL-DB, that will take you to our FTP site. And you will see here that we have all the different files that you can download for ChEMBL-20. So we have a file of all the FASTA files for the protein targets. We have an SD file of the compound. We have a tabbed file for all the activities, etc. You can also download all of the data to an Oracle system or MySQL if you have that instead. All the release notes on how to use these downloads are here, and if you want to have a look at the schema to see how the tables are constructed, then you can click on this picture here. You can also go back and go higher up into ChEMBL-DB and get early releases of ChEMBL should you wish. So now I'm finished with the presentation, but I'm going to do a couple of quick demonstrations on the interface itself just to show you the different things and the different ways you can search. So I'm going to come out of the presentation and I'm going to go here. So this is what the ChEMBL homepage looks like. So I've already drawn a structure into Marvin. I can choose to add to this here and run my substructure search. And you can see this has brought up 797 hits from that substructure. I can then go to the drop-down box to the right-hand side and download everything as an SD file, or I can just go straight to display the activities for all these compounds. Okay, this seems to be taking a little while. I think there must be a lot of activities. So we'll skip that one and we'll go back to the home page while it's still thinking. It does work, but I think it's just being a bit slow because I'm on Wi-Fi. I did mention that you can use this top field to do a lot of searches. So if I wanted to look for hepatotoxicity and click on assays, it will look across all the assay descriptions and the assay names to find any of them that have hepatotoxicity in them. So as you can see, it's brought back 516, and again from here you can still access all the activities linked to these assays, or you can click on a hyperlink and go into the assay report card, which is very similar to the compound report card and target report card, in that you can access all the activities or you can access all the compounds as well for that assay. So I said you can run searches from this field for targets. So I'm going to look for P11229. So you type in your search criteria, click on targets. That will bring you anything related to P11229. So you can see that it's part of a group and a family. You can then go into here, scroll down, still loading. So you can then access all the activities here, click on the grey box, display all records. So this is now all the activities that we have stored for target P11229. And then you can download everything as an Excel file, which you can then store on your own system and mine it for all the information. So you can see how quick that search was, and you have all this information. So going back to the home page, I showed you the browse targets. So this is the hierarchy that you would be faced with, and you can type in your keyword, run your search, and you'll see that I've chosen one that just happens to be quite common, but you can run your keyword search and it will make bold anything that matches that search. If you're interested in something in particular, you can just click straight on the hyperlink and it will bring back everything. You can then choose some of them and display the activities, which, as before, you can then download into a spreadsheet. So it really is that straightforward. I don't want to do too much interface searching, because I think it's something that you can play with by yourself. So what I'm now going to do is I'm going to unmute people, and if you want to ask a question, please will you raise your hand and we will choose people one by one and we'll try to answer your questions for you. Hi Adriana, I saw that you've written that it's too much information. It is a lot to take in. However, what you can do is, I'm just going to go back to my presentation. What we do have is we have a ChEMBL help email for people. So if you do have any questions afterwards, or if there's anything specific you'd like to look for, then what you can do is feel free to email ChEMBL help and we'll try to answer any questions that you have. Okay, so if no one has got any other questions, I'm going to end the webinar. I want to thank everyone for their attention. I know it's been a long half an hour. And as I said, if you do have any questions, then feel free to email us on ChEMBLhelp@ebi.ac.uk. So thank you very much.